Okay, so this is chapter 3. If I can make it go here. Okay, so this chapter, you guys, is all about transport, moving things in and out, specifically of the cell, since this is biology and we're learning about life. So this picture right here is a picture of a what? Okay, so it is a bacterium. It's Bacillus megatherium. Not that it really matters. We could do this for any cell, but what you need to emphasize here is there is a cell membrane. And if you look at this picture in your book, you could see it even closer. You could actually see the line. But you also need to realize that a lot of cells also have something beyond a membrane. It's a cell. What else would go around the cell? A cell wall. Not all of them. If you look at our model right here in our classroom, there is no cell wall. Okay, you could this teal color is showing you the cell membrane. This is a pretty nice model, actually. The teal color everywhere is made out of phosphobilayers. They're membranes. You have membrane-bound organelles in this cell. You do not in the bacteria. The bacteria is much smaller. It's simpler. There's no nucleus. Back to the cell wall stuff. Bacteria has a cell wall. Plants have a cell wall. This does not. What model is this of? What type of cell? What? It's an animal cell. Okay. Now, if you remember your kingdoms, there's 2 kingdoms of bacteria. They have cell walls. Plants, do they have cell walls? Yes. Animals, do they have cell walls? No. And then what other kingdoms are we missing there? Fungus, do you think it has a cell wall? It does. And then there's Protista as your 6th kingdom. Some have a cell wall, some do not. The Protista kingdom's kind of a mess. And there are animal-like ones, fungus-like ones, and plant-like ones. And if whatever one they're more like determines normally if they have a cell wall or not. They have that similar characteristic. Okay, so you need to understand that all cells have a cell membrane. You also need to understand that the cell membrane's number one function is getting things in and out. Do not say that the cell membrane protects the cell. It is flimsy, it is fluid, it does not do that function. You could say that for a cell wall if it has it, okay? You could say that it holds the cell, makes the boundary of the cell and holds it together. But the main thing I want you to remember is that the cell membrane controls what gets in and out, okay? Now, cells need a lot. This is a characteristic of life. So this goes through a whole list of just some nice examples that you need to be familiar with. Of course, we got to get water in and out. And water actually doesn't like the inside of the cell membrane. So it goes through special pores, special proteins just for water. All these ions also have to get through the membrane, and they're charged. That also makes a problem. But like sodium, where do we get our sodium from? Salt, like that in that video, it talked about sodium. Sodium chloride is table salt. Okay. Now we need it, but in much smaller amounts than you saw in your goofy video. The cell needs to get gases in and out, like carbon dioxide and oxygen. Nutrients. We just got done with chapter 2, all about absorbing nutrients. What type of nutrients? What's the simplest molecule that your cells want and need to get? Sugar, that glucose, awesome. Hormones that need to be moved around. Maybe this particular cell makes this certain hormone, this certain chemical. It needs to be transported throughout the body to different cells. They need it in. And then wastes. Underline that in. So you start to recognize that ammonia is a nitrogen-based molecule. We actually have an issue with nitrogen. It is toxic. It is bad for us. We have to get rid of it. Hold on. It's bad for us. We make it as a waste product, and some forms are bad for us. We need nitrogen, though. It's in our DNA, right, in our RNA. But it also is poisonous, and that's what kidney dialysis is all about. Because if you cannot get those bad things out of your body, you will be poisoned. Okay, so moving on. So let's talk about the structure of the membrane. So first, the membrane is made up of phosphates. Do you guys remember what a phospholipid looks like? Do you guys remember that? Do you see this picture here? This is a phospholipid. So it has a round head and it has two fatty acid tails. And I told you the membrane's bilayer, so it has two sides of this. We're going to do a beautiful coloring with it later. So I'm just showing you one of the phospholipids here. Do you guys remember from chapter 1 that the head is hydrophilic? What's that mean? It loves water. What about the fatty acid chains, the tails? They are hydrophobic, so they do not like water. Okay, so they align this way. The other thing is they are made of protein. So I have a model up here, and if I take my model of proteins out, this box of marbles is very much like what the one side of the membrane looks like from the outside. I want you to understand that the membranes are fluid. That means these phospholipid heads can move around and they're not stuck to each other. They like each other, so they're hanging out together. So the other main component is the proteins. Now there's a huge variety of different proteins embedded into the phospholipid layer. So I have these little pieces to represent proteins. Some of them have sticks coming off them. That can represent the carbohydrate chains, for example, that is used for communication. That's in the membrane. You need cell-to-cell recognition. You want cells to communicate with each other. But then also we can have a protein that doesn't have a stick, that maybe it's just a transport protein. Things come in and out of it, like that water. So my little box of marbles is a nice model, because it's still fluid, of a membrane. It can move around. The other thing that you need to remember, and you guys want to write this down, is this is called the fluid mosaic model, because it's fluid. And a mosaic, because what is a mosaic? What is it? Nobody's heard of the word mosaic? You've seen, Oh, like those kaleidoscope things? No, but I could see where they're kind of similar for some reasons.
It's like it has like the virtue of, it's like you know those the art where they like keep like stones and like those plates they smash it and they like stick them together.
Okay, so we have a bunch of different things. They could be broken and smashed.
It's like a piece of work made out of tile.
Okay, so we have an end product, which is a piece of art, and it normally makes into some picture, right? Ones I'm more familiar with have to see, like I remember a shell one. So here we have a variety of different things, like you got phospholipids and you got different types of proteins. You have a variety of different things that come together to make something that works. A picture, say, of a shell, right? And there's a lot of awesome ones down in Mexico, right, where they use the broken tiles and stuff and make these beautiful pieces of art, these beautiful pictures. So can you see the analogy here, that here you have a variety, and I went ahead and put these little wiggly things in here in my model. That can be like cholesterol too. Cholesterol's in animals' membranes. And the cholesterol makes it even more fluid, so it's not rigid. But anyway, the deal is we have a variety of things coming together to make one working product. Just like all those different colors pieced together make a working picture. Or, you could see the picture in it. So this is a fluid mosaic model, this cell membrane. This last line here you might want to underline, really important. It says usually the polarity, the size, the electrical charges of molecules determine whether they can pass through the membrane. So it is very important that you guys realize that the membrane selects what can come in and what can come out. So that's known as selectively permeable. A screen on your door or your window is selectively permeable, right? Only certain things can get through it. So is the cell membrane. Okay, so let's watch this and listen.
Not all molecules are equally soluble in a membrane. The nonpolar phospholipid tails of a lipid bilayer tend to repel charged particles such as ions, but allow fat-soluble molecules to pass. Usually, the polarity, size, and electric charge of molecules determine whether they can pass through a membrane. The small molecules of nonpolar gases such as nitrogen and oxygen can pass through freely. Small polar molecules such as water, glycerol, and ethanol can also pass freely across the membrane. Larger polar molecules and ions would be repelled by the membrane. By limiting entry, a membrane is selectively permeable, which means that membranes regulate the exchange of materials in a very specific way.
So you guys have to remember, this reminds me with your harder test questions. If I said glucose, can glucose get through a semi-permeable membrane? Can it get through the membrane just through the phospholipids? Can it? No. Why? It's too big. So if I gave you another big molecule, you'd have to be able to reason that out. Okay? So some things can fit through easily, some things can't. Some things need help in the form of a protein. So that's these transport proteins. That's those little things you saw that I jammed in between the marbles there. Okay, so some proteins allow things through that can't normally get through. Now, are proteins specifically shaped? Do proteins have a specific shape to them? Like the enzymes, do they? They definitely do. They do. And it's the same thing here with this. These proteins are shaped perfectly to allow the things in and out that they're made for. Okay, so these are transport proteins. I mentioned the selectively permeable thing already. Now, I'm glad you brought that up, because when a protein is just there, right, it's ready to do its job, it has a perfect shape. Sometimes, depending on when it's doing its job, when the other thing comes through it, or like with an enzyme it attaches to it, it can like change the shape a little bit, but not much, but a little bit. And then, maybe it releases it, or it goes through, or whatnot. But if a protein's in a bad environment for it, too hot, too cold, too acidic, it falls apart. It's never going to get folded back like it's supposed to again. Okay? So shape is extremely important. Okay, back to all the different things that are in the membrane. The glycoproteins are the ones that have the carbohydrate chain attached to them. What are carbohydrates? Yeah, sugars, glyco. Do you see that? Does it remind you of a word? Does it remind you? Glucose. So let's try and remember that. So these ones are the proteins that have a carbohydrate chain attached. Now there's also glycolipids. Is that a protein? Glycolipids. No. So there are some other things in there. This is a sugar attached to a fat. Okay? And there are and a lot of these things act as those communication tools for the cells. The cells recognize each other and they talk to each other. Okay, let's check this one out.
Membranes are composed of a lipid bilayer and proteins. The bilayer of phospholipids provides fluidity. Some proteins that are embedded in membranes have sugars attached to them and are called glycoproteins. Sugars also can be attached to the heads of membrane lipids, creating glycolipids. Glycoproteins and glycolipids act as antennae that receive chemical messages from other cells. Proteins extending through the bilayer or embedded in the membrane's inner or outer surface make the surfaces asymmetric. Carbohydrate chains may be attached to both lipids and proteins. Membranes in animal cells also contain cholesterol.
If you're curious about that word asymmetric, it means it does not have symmetry. So that means it does not, one side can be different than the other side. That's simply what it's saying. It's a little more advanced. I don't really care that you know that. but I just wanted, since he used the word. Now notice these cholesterols. Do you see them there? They're very globular looking, right, in this particular model. These are models. I made my cholesterols in this one more chainy looking. When you guys do your coloring, the cholesterols are attached to a little round thing. It's not as big as, it's not a phospholipid head. So when you guys do get your coloring, make sure you see the cholesterol in here also, okay, because it looks different than the model in your book. It just depends how the author decides to draw it, how well they try and represent it to reality or not. Okay, also with the coloring, I want you to notice right now that he said there's two different types of proteins. Some are peripheral, meaning like peripheral vision. What? It's just on one side. That's not going to be a transport protein, is it? Now, an integral protein is fully integrated, right? And so it goes all the way through. So is a transport protein, is it an integral protein? Is it a type of integral protein? Yes. Okay? So once again, if you guys remember before, proteins have so many different functions. There's just so much variety of proteins out there. And you can see that in your cell. Okay. Stay with me. Okay, we got a big bulk of this done. I got to do another few slides and then we'll be done for today. Okay, this is also hugely important. So now, how do atoms and molecules, how do they move on their own? What's going on? And that's called diffusion. Okay, now diffusion is a passive type of transport. What do you think that means? Passive. No energy is required. It happens on its own. It happens from the energy of just movement. Atoms are always moving, and things slowly spread out. So look up here, people. Do you guys see this dark violet red spot right here? Can you guys see that? Can you guys see my arrow? So what happens here is this is really concentrated with these violet molecules. These are these pigments. They happen to have color so we can see them. We drop this drop in the distilled water. Those pink molecules start diffusing. They go from an area of high concentration to an area of lower concentration. And the molecules, just by randomly banging into each other, over time, it gets consistently dispersed. This is at equilibrium. Now, this is the important part because there's a lot of words that you could use here to talk about this stuff. This C cup is in equilibrium. There is no concentration gradient. A concentration gradient is when there's a difference. Gradients, grades. Is there a difference between 9th grade and 12th grade? That is the biggest concentration gradient we have in high school. Compared to, say, freshmen and sophomores. There's less of a difference between those particular grades. The graduated cylinder has grades that account for different amounts in here. A steep road, if you guys do some construction and maybe you've moved dirt in your yard, or you hike, you're going up a grade or down a grade. So concentration gradient, there is a what? Difference. Okay? And stuff, energies around, things are randomly, things will go downhill, right, without added energy, and things will diffuse on their own, passive transport. Okay? Now, of course, life takes advantage of these things. So I talked to you about concentration gradients again. Diffusion is passive. The molecules will move from a high to low. That means they're moving with the concentration gradient. Now, osmosis is a type of diffusion. It's diffusion of what?
Water.
Beautiful, water. Okay, that's all it is. Water diffuses, or you could say osmosis happens. And you know that water's diffusing. Okay? Now, does it take energy? No, because it is passive. It happens on its own. The last vocab here on this slide is turgor. Plants take advantage of this more because they have cell walls. They want water coming into their cells. They want water to help them stand up straighter. It gives them pressure. It gives them structure to be full. Okay? Now, animals don't want this because they will break, split, because they don't have a cell wall. So this pressure is called turgor that animals have. I will show you a picture in a minute. Now, diffusion is how fast diffusion happens, the rate of diffusion, is affected by how big the concentration is, how much volume is there. Okay? So if one of you guys just let out a nasty old fart right now, and they're in the back of the room, I know you guys in the back of the room, you guys are the ones farting all the time, it's going to take me a while to maybe sense the smell, because those fart molecules, those methane gas molecules will diffuse. Poor Paul over there, he's going to get blasted right away. Okay? So, and you guys have noticed it more often, I hope, than farts when people are spraying perfume in the classroom. Right? And it sucks if you're sitting right next to the person. It might be pleasant if you're sitting a nice distance away because the diffusion occurs. Okay, so now, really focus. Here's the example. So in this one right here, you could see the iso right here. I want you guys to realize that these cells are in an isotonic solution. Write it down. Isotonic solution. ISO and tonic, T-O-N-I-C. That means there is no concentration gradient. It is the same on both sides. Is the animal cell happy? Yes. Sure, things are coming in and things are coming out, but it's not going to make it change size. What about the plant? It would rather have water coming in because it's not getting that extra pressure. That's an isotonic solution. Okay, this one, you have high water concentration. Now, this might seem backwards. You have high water concentration, but that means you have low solute concentration. But osmosis is about the water. So let's look at the perspective from the water. Now, first, I want you guys to know that this is called a hypotonic solution, right? H-Y-P-O and tonic, meaning that there's not that many things dissolved in the water. So where is there more water then? Outside the cell. So water moves from a high concentration area of water to a low concentration of water. So where does the water go? Into the, we're going to go from high water into, it's going to diffuse, so it's going to plump up the cell. We have a cell wall, so no worries. The cell wall will hold that pressure. But what about the red blood cell if it's going to put a lot in there? It's going to burst. Now the last one here, oh, no, not that. No. Is not hypotonic, it is not hypo, it's called hypertonic. Now, how I keep this straight in my brain is I memorize the hypertonic one because hyper means busy, active, right, to me. So then there's a lot of stuff dissolved in the water, but that means you have a low water concentration, hypertonic. What happens to the cell? It shrivels because the water is going, it's too salty, for example, the water, so the water leaves the cell because there's a lot of water in here, goes out. Does this make either one of these types of cells happy? No. And this is what you're going to see on the lab for your onion. We're going to use onion cells, and if you do this right, you should see that cell membrane come off the cell wall. That's not good. The cell's going to die. Okay, so you want to look for that when we do the lab. So this is, one more time, what type of solution is this? Hypertonic. What type of solution is this? Hypo. And here? Beautiful. Okay, so now we're almost done. Hang in there. I have already mentioned to you passive. What do you think active is? Requires energy. Now hold on. We are still on passive when we get to this facilitated diffusion. So maybe bold the passive. Okay? So facilitated diffusion is just simple diffusion, except we have a helper. That's where the word facilitated comes in. So we need what to help us get this thing to diffuse in and out of the cell? We need a transport protein. So this is like an open door, for example. So now we got people spraying perfume up here. And if this door, forget I opened it, let's just say it's always open, okay? Can things move back and forth from the hallway to the room? Because of this special transport protein, right? For in our cell. So is this passive or active? It's still passive, okay? But the cell has this special opening so these particular things can get in and out. So this is still of diffusion, still passive. It's facilitated because what is necessary? A transport protein. Beautiful. Okay, almost, people. So this goes over the two definitions. Now notice the different wording. Active transport requires what? Energy. We got to add energy. I could also quiz you and say that this moves things against the concentration gradient. Is that still active transport? So see how well we have to know things to be successful in here. Okay, so moving on, let's watch these.
In simple diffusion, a type of passive transport, molecules pass directly through a membrane down their concentration gradient without any input of energy. In facilitated diffusion, also a type of passive transport, membrane proteins transport molecules down their concentration gradient without energy input. Active transport requires energy, such as from the hydrolysis of ATP, as well as transport proteins. Active transport can move molecules against or with the concentration gradient.
So with this active one, you see it needs energy. What's the molecule that's changing to release the energy? ATP, a phosphate's breaking off. So this takes energy. So is it passive or active? Active. Now you can see they've done a good job in this model showing that the protein does change shape. And if the protein changes shape to that degree, is it going to take energy? Yes. Okay? So that's active. And then look at the concentration gradient. More green things over here. They are diffusing with the concentration gradient. Over here, do you see that they're going against the concentration gradient, but they're still getting more of what they want? Okay, so that's active. Okay, one more topic is this is definitely active. Maybe highlight it so the titles don't confuse you. This is endocytosis or exocytosis. What does cyto mean?
Cell.
Beautiful, cell. Endo, stuff coming into the cell. Exo, things going. Okay, now this is crazy. It totally needs energy because we're going to make more membranes. We're going to change the shape of membranes. So on the A side, you could see endocytosis. That cell membrane is actually growing. Basically, it's not alive. More chemicals, more phospholipids are being attached, and a membrane is being made around something the cell wants. Then it can disconnect, and now we have a vesicle. We have a membrane enclosed pouch of what the cell wants. We can break it down and get what we need inside the cell. Is that going to take energy? Definitely. So it's active transport. Can we go the opposite way, where forget A, just look at B, the cell encloses, it makes a membrane around something it doesn't want. It moves that vesicle to the membrane. That membrane becomes part of the cell membrane, and then it pushes out. Okay? So that's endocytosis and exocytosis. And I think we're going to stop right there.